Hi everyone. Today discuss about lysine. First of all, say about the lysine. The L-lysine is essential amino acid as it is not synthesized by human and seen as B2 from the diet. Lysine is a widely used nutrition supplement in food and behavior industrial as well as chemical intermediate where it is lacking. Microorganisms are capable to synthesize survival amino acid. Some of them is excess of our need. Those amino acids are excreted out the cell and this fact can be exploited in industrial production of amino acid. Next of all, the microbial fermentation leads to the fermented of isomer of amino acid, which is the biological activity whereas the chemical synthesis produce mixture of LD ferment which is need the separate by expensive process. Recent method of commercial production of lysine based on fermentation of carbohydrate. Total whole production lysine is around 35,000 material ton per year. Next of all, see about the history. In 1889, lysine was the first isolation of casein. In 1958, Kisosis and co-workers first report the microbial fermentation of L-lysine using homorysine axotrophic strain, Corynebacterium glutaminum. Hemoserum, also called as isothermic, is a amino acid. An microorganism that it is able to synthesize one of the more essential growth factors and it is not grow fermentation media locking them. Commercial production of lysine was done by using Corynebacterium glutaminum. Then the Corynebacterium glutaminum was used logical production of lysine. Other groups of microorganisms involved in lysine production Bacterium flavum, Bacterium lactofermentant microorganism Glutamix, Arobacterium azosinum, Bacillus lichenophemiums. Then industrial production of lysine, the two types, indirect fermentation and direct fermentation. The first of all, say about the indirect fermentation. Indirect fermentation is also called as dual fermentation. As the two different microorganisms are employed is the fermentation process. Mutagen E. coli is used first of the fermentation and wild types of prototypic Escherichia coli or Agrobacterium employed in the second of the fermentation. From glycerol diamine acid DAP produced in the first step of fermentation by Axotraphia, the Escherichia coli is the converted into lysine by Agrobacteria and second the fermentation. Then direct fermentation. L-lysine can be also fermentably produced from any of the substrate directly and the process is called as direct fermentation. Direct fermentation process the presently employed throughout the world production L-lysine. This is the method of use of the precursor in the biochemical pathway leading from aspartame to lysine can feedback inhibit activity to the enzyme casein axis of the lysine production. Then fermentation condition, aerobic condition, continuous oxygen supply. The carbon source and nitrogen source growth factor. First of all, the carbon source, sugarcane malt source, ethanol with soybean hydrolysis. Nitrogen source is urea and glucose is ammonia or ammonium salts. Growth factor is L homocerium or L theronine and L methionine biofilm greater than 30 UL. Then fermenters sterilization using steam temperature is maintained at 80 to 20 to 13 degrees Celsius. pH is maintained for 7. Then during fermentation, lysine production start early large phase growth and stationary phase and maximum production 40 to 45 lysine achieved 60 years. Yield 30 to 40 percentage of lysine as monohydrochloride reaction to the inactive sugar condition. The lysine is recovered by acidification of the cell-free fermentation broth. pH is 2 within hydrochloric acid. This is the acidophile mixture is then passes through condition exists in column where lysine get ammonium from. Bound L lysine is the eluted from the column within dilute amino solution. The olivary acid and cytosis L lysine hydrolyte. Then finally use of lysine. Essential amino acid required from the human nutrition. Improved nutrient in human from animal. The largest commercial use for growth, purity and other animal feed